start the slideshow.
Mhm.
Okay, can you see my screen?
Yeah, sure.
So, I believe we can start. Hello, dear colleagues. My name is Alla, and today Valeria and I gonna present you the paper called Spades in Genome Assembly Algorithm and its Applications to Single-Sex Sequencing. So, basically, what is the motivation of this paper? Most bacteria in various environments cannot be cloned in the laboratory and therefore cannot be sequenced using NGS technologies. But single-cell sequencing is an alternative to collecting a large number of genomes, and it could be profitable to use this technology, for example, for human microbiome project. Single-cell sequencing can capture a large number of genes sufficient for inferring the organism's metabolism. Particularly, this fact can be useful for such projects as human microbiome project. And in many applications, having a majority of genes captured is as useful as having incomplete genomes. But single-cell sequencing is highly erroneous, and we need to come up with an algorithms and approach to fix it, to enhance the quality of single-cell sequences using algorithmic and mathematical approaches. So let's briefly dive into graph theory. All the considered graphs that I'm going to discuss are directed ones. Vertex preceding is a definition when vertex V in graph G have an edge from W to V. In degree and out degree, correspondingly, a number of vertices preceding and following V. A vertex V in graph G is called hub if in degree doesn't equal to 1 or out degree of its vertex doesn't equal to 1. For example, these ones are denoted by arrows. These vertices are hub ones. A directed path in graph is called hub path if its starting and ending vertices are hub nodes and its intermediate vertices are not hub nodes. An edge is called h-edge if it starts a hub. So it's basically first edge in the hub. Given an h alpha, we define the h path starting as a path alpha and denote the number of degrees in this h path as this notation. If a in the ith degree is h path starting from an h alpha, we define h at from a as alpha, and offset of a is I. So basically, it's position of vertex a in the hub path. Also, authors of the paper use the notation of de Bruijn graph. Let's briefly describe what is it. N-mer is a string of length n, and given an n-mer a, we define a prefix like a first part of the string with all of the nucleotides except the last one, and suffix of a is all parts of the strings except the first one. For example, ACCG is a 5-mer, and for this 5-mer prefix of its prefix is ACC and suffix is CCG. And for a set of strings, let n be the number of chimeras that occur in string and reads as substrings. And what is the definition of the de Bruijn graph? Initial graph G0 on form on 2 n vertices. For each chimera that occurs in strings and reads as a substring corresponds to vertices u and v and form an edge u and v. Label of the edge is of hu is prefix of a and v is suffix of a, and edges are labeled by k-mers and vertices by k-1 mers. And then we glue vertices of G0 together if they have the same label. So basically, we have vertices with k-1 strings and edges with k, denoted as k string of this k-mer. And in the de Bruijn graph, h path passing through n vertices defined as n plus k minus 2 mer and called a hub read, substituting every h path in de Bruijn graph by a single h labeled by its h read results in condensed de Bruijn graph. reads at K called the set of all H reads in de Bruijn graph, and coverage of an H is of the de Bruijn graph is the number of reads that contain the corresponding K-mer. The coverage of a path defined as the average coverage of its edges. So if we have an H and there are some reads that correspond to this H, we define this number of the coverage as number of reads that fall into this H, and the coverage of the H path is the mean coverage of all these edges that persist in this H path. Also, author of the paper introduced the notation of multi-size de Bruijn graph. Why is it so? Because the choice of K affects the construction of de Bruijn graph. Smaller values of K collapse more repeats together, making the graph more tangled. For example, this graph is very tangled, so there are a lot of paths, and it's hard to deal with such graphs. And in opposite way, larger values of K may fail to detect overlaps between reads, particularly in low coverage regions, making the graph more fragmented. For example, in this picture, you can see a more fragmented graph with a large number of K, where there are a lot of not connected components in this graph. And it's also highly problematic to deal with this graph with standard algorithms. And therefore, we introduce a multi-size de Bruijn graph that combines the advantages of both low values of K and large values of K. And the definition is the following. Given a positive integer d less than K, we define reads K minus d, K as the union of d plus 1 sets. Reads K union reads K minus 1, and so on, to K minus d. The multi-size de Bruijn graph is defined as de Bruijn graph K minus d, K, and K. So this is an example of multi-size de Bruijn graph. You can see that there are a large part of this graph and the smaller ones, and it's neither tangled nor fragmented because we used the longer path with high coverage in this region and shorter path in the lower coverage, and it combined in a more prettier graph. So let's discuss the stages of algorithm itself. First stage is assembly graph constructions. So on this stage, we basically iterating over all list of values K0 less than K1 and so on to T by constructing the following graph, de Bruijn graph of reads from K1 or K0 to Ki is de Bruijn graph of reads union H reads of dp reads K0, Ky minus 1, and Ky. H reads from the graph is the set of H reads associated with all H paths in G. Also, this step include error corrections. Despite multi-size de Bruijn graph improve standard de Bruijn graphs, we still need to correct so-called bulges and tips. For example, bulges is depicted on this picture, where we have two alternative paths, and we need to choose between them because we need to have a full graph with no alternative or doubled way. Tip is also two or more alternative paths, but in case of tip, we have a path that is not properly starting or, for example, ending. Also, there are chimeric reads. It's also a way of alternative paths, but with different topological conditions, and repeat is another example of errors. Bulge tips and chimeric reads are detecting in the Spades algorithm using the topological properties, for example, out and integers, as I described, and their relative lengths to adapt for properties on different SCSC datasets, and relative mean coverage. Relative also in terms of relation between P and Q, which are alternative paths. Tips and chimeric reads in Spades are removed, But bulges are not removed like in usual algorithms, but they are co-removed. They are corrected and removed. So removing path from P from the graph and substitute each H of P by an H projection in Q is operation described by this formula. So basically we calculate the relative position of nucleotide that does not correspond to Q into Q path, calculating the relative position of Q and P and its offset on the P. Also, Spades maintains merge find set data structure for storing logs of graph simplification steps. It's crucial for the first stage when we need to reconstruct contigs. And basically, this data structures allow to map the original reads for the resulted assembly graph. Spades iterates through all of the list of H-paths in increasing order of coverage for bulge removal and chimeric H-path removal, or iterates in increasing order of length for tip removal. It updates the list of H-paths that were deleted or projected, and Spades rescue more H-paths in the graph by the following properties. By safer than removal projection operation that I mentioned already, using topological feature of the graph instead of using just lenses and coverage like it was in previous algorithms, and more accurate gradual path removing. So basically the iterates of all paths in increasing or decreasing order of the coverage, and all three properties allows us to reconstruct it more accurately. So the next stage is stage 2, utilization of read pairs in Spades. The genomic distance between a pair of H pairs can be estimated when they are linked by reads. The estimated genomic distances between the two reads in BiRead are aggregated over all BiRead connecting a given pair of H paths through a series of the following transformation depicted on this image. So let's split it up for stage by stage. B transformations transform BiReads into K-mirrors, a pair of edges in the deep graph, which we refer to as bi-edge. So BiReads transform to K-bimers, or bi-edge. H transformation, given a bi-edge A, B, and the estimated distance D between these edges, we define H by H and A, B, D as H of A and H of B. So basically, as you recall, these are the starting edges of the path that A and B belongs to. And the most crucial part is D, is a new estimated distance, which equal to the D plus offset A and minus offset B. Why we need to do so? Because D as a distance estimate is very inaccurate in original genome data, and we need somehow to aggregate and reconstruct the original distance between the edges. And the A transformation do the following. In H-page histogram of multi-set of H pages, we fixed H edges A and B in its variable distance estimates. The peaks in the smooth histogram obtained by fast Fourier transform will approximate the actual distance in the adjusted H A by H, A, B, and D, or a small number of adjusted H by edges. So for example, if we go to the simpler terms, we have a lot of different estimations of distance between passes, and we aggregate them using a histogram, smooth it, and choose only one peak or maybe two peaks in case of there are two peaks presented on such histograms, and define the original, the statistically more popular distance between these passes. And the E transformation do the following. Oh, I'm sorry. Given H page A, B, D, we split this H page into the set of bi edges such that A belongs to pass A and B belongs to pass B, and D plus offset A and offset B is D. So basically we choose the D that is more probable, and we only leave such edges that belongs to the paths that have more probable distance. So the next stage is the stage 3, and my colleague gonna present it to you.
Hello, everyone. Stage 3 is from PDB-JS to paired assembly graphs, which consistent LRN cycles. So C is a known cycle in a multigraph G. We want to find it to construct our genome. We defined the BH as a triple A, B, and D, where A and B are adjacent G, and D is an estimated distance between A and B in the cycle. We define start, end, and as a pair of vertices, and a cycle C is consistent with this triple BH if there exist instances of edges A and B at the distance D in C. And given a set B of BHs, our cycle C is B consistent if it is consistent with all BHs in BE. And now we can define the BH consistent LRN cycle problem. So with given an LRN multigraph G and a set of BHs B, we need to find a B consistent LRN cycle in G. So how do we find it? We first need to construct BH graph on a set of adjusted BHs. So we use some algorithms, and to construct it, we define an initial graph G0 on to be vertices for each BH. Introduce two new vertices U and V, and H from U to V. Label the results in H by BH, label U by vertices start, and V by end. So we had circle string genome. We have BH set BE. It's by Merz-Caddy genome. That's it is a set of all K bimers from genome separated by exact distance D, and we have graph DB, and BH consistent LRN cycle problem is defined by the graph DB, and the BH set BE. Our genome, our string genome, defines a B consistent LRN cycle in DB, and thus represents a solution of this BC-EC problem. Next, we construct graph DB as it is shown on this slide from our genome. So we define first ABD as a BH with minimal offsets among all BHs in E, and for a given HBH, all edges in the BH graph labeled by BHs from E. Well, this from a subpath of an H path, and this subpath starts with first and ends with last, and they do not share any edges. For let's look at this example. At this yes, thank you. This graph, let's look at it. It has 4 hubs, 4 vertices marked with red color, and it has 4. It has 6 paths, each path is from first to 6. It marked with alpha first, alpha second, and so on. So alpha first has only 1 edge, so it is first, then second has 2 edges, third 3, and so on. And to go through this, all of this edges, we can use the following order as it is written on the slide, and note that we go through P1 and P2 twice, because we included some vertices, and so now it is happened like that. Then next slide. So we, with known in advanced genome, we can measure exact genome distances. Each read from genome contributes information about genomic distance that is collected on HBH. For example, let's look at the by read ACGTCA, AAGTTC. You can find it on the graph. It has 4 bimers at the distance 5, and it gives us 3 HBHs, as it is written on the slide. And with all of the byreads, we can construct all HBHs. So on the slide, you can see 13 HBHs, and they, next slide. If we work with them and we put them on the area, and well, with these rectangles, I should say that if you are very interested in the exact way how we construct this area with rectangles, you should read the paper, because we don't have time to go deeply into details. But our main goal is to find this blue line. It is straight, and it is well, have 45 degrees, so we need to find it. But the problem is that in real life we don't know every distance, every exact distance. So next slide, please. We should work with distance estimate error delta. And this is how we modify the previous algorithms. So now we work with multi-label graph. And now our rules is H1, define an initial graph G0 onto HBE vertices. For each HBH, introduce 2 new vertices, as we had in the previous algorithm. But now our labels multi-labels. So multi-label U by a set of bivertices start set D, and multi-label V by a set of bivertices end set D. And H second glue vertices of G0 together whenever they share at least one label. So our labels don't need to be the same with all of them, but just one, and we glue them. Next slide. Okay, and the last stage is backtracking edges relocated during the graph simplification. After all stages, stage 4 restore the position of reads along the path using the information about reads stored in the data structure mentioned on the stage 1, and displays final contigs.
So finally, let's switch to the results. As you can see from the table of these results, Spades improve state-of-the-art algorithms for single-cell sequencing based on a large number of metrics, for example, number of contigs, number of base pair, and most importantly, covered percent of the genome. Spades is moreover applicable to standard datasets too, not only to single-cell sequences, but to the usual ones as well. And single-cell sequences, fragment assembly has a great promise. The potential of NGS data for single-cell sequencing has not yet fully utilized. So this is the end of our presentation. We know that it was rich on formulas and notations, and we would be happy to answer your questions, or maybe to explain something in the details. You are welcome into the discussion with us.
Hi, thank you for your presentation. I have a question. In the almost in the beginning of the your presentation, you explained how the length of the сейчас еще, наверное.
Нет, это оно.
Or is this the start? Oh, so can you go a little bit further?
Okay. The
No.
choice of
I mean the presentation. Just and a little bit. And a little bit also. Okay. Well, I unfortunately don't remember where is it. It was specifically.
But what is your question?
Yeah, you explained that the length of the path, not path, but H graph. I remember you have 4 nucleotides, and you said that the length is 5. For example, I still don't really.
I'm sorry.
Oh,
This
yeah,
is typo.
here.
It is 5. Oh, it is not a 5 mirror. It's a 4
Okay.
mirror. And basically, what I wanted to say is that H is a full k-mirror, and prefixes are vertices, and they are truncated version of this k-mirror from the beginning and from the end. So this is the purpose of this example. I'm sorry for the typo.
No, that's okay. Thank you.
So no more questions, I guess.
I have one, if you don't mind. How I understand this algorithm tries to assemble a linear chromosome.
No, it's not a linear chromosome. We talked about a bacteria genome from the very beginning.
Okay. It's all because I think the graph tried to be drawn like a circle when you.
No, it's a circular genome, and this is why we have all the illustrations with circular genomes.
Okay, it's all. Thank you.
Yes, you can see the genome. It is written as a cycle.
So,
All right.
now,
I guess we don't have time for
okay.
We are
questions.
really happy that you could listen to this hard presentation. So thank you very much.
All right. So let's go to the next one. I guess this one was the most technical of them all. I get some of them are less, I don't know, notation heavy. So maybe easier to listen to.
All right, can you hear me?
Yeah, sure. You can see a screen also.
The brain graph breaks in again, so to speak, and we'll see it from a little another side, and watch how the brain graph can handle long reads. So actually, we have 2 strategies to produce long reads from machine, sequence machine, or short reads. And there's the pros and cons of each strategy. For instance, if you produce long reads, there's a better chance to cover the part of the genome which have, for instance, it's highly polymorphic, or there's much repetitions, or some complicated mutations which are hard to map using short reads. But at the same time, there is mostly use Pacific Biosciences technology to produce long reads, but due to some technical issues, biological things, long reads are prone to errors. At the same time, if we use, for instance, Illumina sequencing, we can produce shorter reads, but they are more accurate, and the quality of the assembly apparently is better. But here's the solutions to how to map and assembly the long reads. Prior to the Berlin graph was used overlap level consensus assembly, OLC in short, and it's literally matches the all reads against all to find the overlap and apparently construct the contigs and apparently the genome. But as you can assume, it's very time consuming because you compare million of reads and it takes many time to do it. The Berlin graph going another way, it's use a graph to reconstruct the genome, and there are some misconceptions that the Berlin graph can be applied to long reads because actually it's true that the Berlin graph was used to assembly the short Sanger, long Sanger reads, but they are very accurate, but it's not applicable to long error-prone reads. And the authors of this paper suggest how we do nowadays. But before we go deep of Berlin algorithm, which is a variation of the Berlin, let's get into the key idea and how the Berlin works. It's take some compositions. For example, it would be read, and it divided in the chunks using the conceptions of k-mers, which is some number of how we divide our chunk into the some others. And for instance, we use KS3 and take possible k-mers. For instance, we get TAA, AAT, ATG, and etc. So we got to the end. Then we construct our actually our vertices of our graph, and we do it merely. We from each k-mer we produce two k-1 nodes. For instance, TA, AA, AAAT, and etc. And it's actually a reason why you can't actually set the not odd number in VK if you use clustering or some other algorithms, because it should have the center to produce the k-1 mers. So we build up our nodes, and we find the overlaps between these nodes. For instance, we see these nodes actually are the same. We match them and produce the H. We go through, find the other ones, traverse to the graph, and glue them again. And I will skip the other steps, and apparently we get the whole picture how graph looks like. And you can notice that these edges, it's actually our initial k-mers. So when we do the same, we find our nodes and conjugate them together physically, as you can see this part and this part of the graph are conjugate together to glue them again. Here's the circle one. glue them together, traverse through, find another nodes, conjugate them together and glue them. And apparently we get such a graph from which we actually can reconstruct the initial genome. And let's begin to the Abraham graph, which is the main tactics algorithms which apply to the long reads, and which the difference. We can see the same picture graph because it's probably the genome of the bacteria, and there's the same tactics to match the nodes and glue them together, but where in Abraham graph we can use, so to speak, substrings-free nodes, meaning that we can use not k-1 nodes, but we can use a whole k-mers. And there's a special formulation for such k-mers. It's a solid string. Solid string means that it's most frequent in the genome k-mer, which can be representative for constructing the graph. But how we select them? We cannot indeed randomly. So we should define how which number of repetitions of such there that it's occurs in the genome only once. Repeated if it's occurs multiple times, and non-genomics k-mers, it's which are just probably the error of the sequence machine, which cannot be non-genomic, can be genomic for a building graph. So authors figure out that selecting a smaller than larger k result in slightly better performance. And it's still we talking about the solid strings, which are frequent k-mers in the genome. And when there's an issue, we should balance between the amount of k, because if a string consists of all k-mers, and it may be easier to tangle in a graph, and it's hard to represent some chunks of the genome if a k is small, and it can be too fragmented if the k is large. So there's some regulations about the k, value of k, and how much it's occurring in the genome. And we should proceed to the how its graphs are built. So actually, it's very complicated, and there are some conceptions, but I omitted it in order to preserve the time. But main point about that it actually creates parallelly many other graphs on the same chunk of the genome. And we can see the blue lines and red lines. It's alternative graphs which can go upward to the from 5' to the 3', and go left from and vice versa. So why it's so? It is used as important metrics to verify the certain chunk, certain counting, for instance, to be sure that it is looks like exactly to build the main graph, which will be representative of some chunk. I know it sounds complicated, but I hope it will be more comprehensive on the next slide. So as it was mentioned, the previous performance, there can be bubbles and tips. And bubbles means that we have a divergence, and it's when we meet some polymorphic sites or repetitive ones, and it's a more problematic chunks of genome which we can't say for sure how it's looked like. And here's the Hilbert Spates, a model of the Spates assembler, and it uses the short reads and long reads altogether. For instance, it was using the Oxford nanopore, but there is also long Spates which use the conception of Hilbert Spates, but there is only a difference that on input it takes only long reads, and it uses the graph, builds up many graphs. For instance, if we go upward and at the same at certain time it holds, we can build up a backward graph to verify, to support some occurrences, some contigs, because Abreu maps all reads to constructed contigs and uses the remaining reads to iteratively construct other contigs. And by that, it attempts to find the major graph, which can be representative of the some part of the genome. So, Katerina.
Oh,
My colleague.
is your microphone not working, or? I mean, if Ekaterina is trying to say something, we cannot hear her.
Oh, sorry. I begin this talk, but sorry.
Yeah.
Okay, yeah. After this, we need the purpose of the correction error draft. And the first step, it is defining the multiply read alignment of the entire draft genome into thousands of short segments, and they called mini aligned segments. Second, after this, error correction of each segments into a consistent mini aligned malign. Bruchin use Blazar. It is bioinformatic tools which used to align long DNA sequences, and it aligned reads against the draft genome. After this, combines pairwise alignments of all reads into a multiplying agent. Important that many segments in Blazar alignments are not aligned, so the accuracy of our error correction approach may deteriorate. So we need a good partition on the draft genome that satisfied two criteria. First, it is most segments in the partition are short, so that the algorithm to their error correction is fast. Second, the region of the read aligned to a given segment in the partition represents an error phone version of this segment. We refer to a position of the line. Okay, sorry. Yeah, okay. We refer to a position of this line meant with space symbols, space in the reference sequences as a non-reference position, and to all other position as reference position. Next slide, please. Thank you. The column in multiplying alignment containing the is positions and in a given regions of this references genome is column for this position. Cov of E is equal match plus sub plus del. And we define the match substitutions and the search arrays at position E as divide all match substitutions and deletion insertions, they are all divided to cov, given EMS in draft genome, we define its local match rate at the minimum match rate among the positions within EMR. The first then define its local insertion rate as the maximum insertion rate among with the position with this EMR. Breaking the next slide, please. Yeah. Alpha, beta, solid EMS are accurate, so they use construct yet another Broadgen graph with muscular simpler bubbles. Broadgen analysis each mini alignment and corrects error each segment between consecutive landmarks. We refer to the multiply alignments of all reads against draft genomes as alignment, and we label each landmark by its landmark position in alignment and break each read into a sequence of segments aligned between consecutive landmarks. Further, each read represented as directed path through verticals corresponding to the landmarks, and it spans over. Near you can see, no, not the slide, earlier. Yeah. Okay, yeah. Label, yes, okay. We refer to the multiplying alignment, the draft genome as alignment, and we label each landmark by landmark's position in alignment and break each read into a sequence of segments aligned between consecutive landmarks. We further represent each read as a directed path through the vertices corresponding to the landmarks that spans over to construct our Broadgen graphs AB. We glue all individually labeled verticals in the set of paths resulting from the reads. Labeling vertices by the positions in the draft genome disguised as identical landmarks from different regions of the genome prevents excessive gluing of vertices in the Broadgen graph AB. As you can see, also there are many bubbles in the graph, each and these bubbles is more simply and make the error correction step fast and accurate. The edges between two consecutive landmarks form a necklace constituting segments from different reads that align to the region flanked by these landmarks. Below, we described how A-Breeze construct a consensus for each necklace. It can call to ELSA the necklace consensus and transforms the inaccurate draft genome for dataset into a polymerase A. Each necklace contains read segments, and the task is to find consequence sequence. The match mismatch assertions and deletion rates should be derived using an alignment of any set of reads to any reference genome. Bridgen selects the segments of median length from each necklace and internally checks where the consensus sequence for each necklace can be improved by introduce a single mutation in select segment. If this exists a mutation that increased the probability of generation segment seg-E, we select mutations that results in the maximum increase and iterate until the converge. We further output the final sequence as the error-corrected sequence of the necklace. Yeah, please, next slide. Yeah. Abrogen collects data set into contigs, maintain structural concordance with reference genomes, and analyzes relative branches assembly to be more complete, missing favor correlations compared with another samples. Next. Okay, a Broadchain algorithm is designed for assembling genomes using long airphone reads, and it is often using for bacterial genomes. Broadchain suggested the possibility of further improving assembly accuracy and category by initial sampling with a Broadchain, constructing the Broadchain graph of resulting contigs, and performing the Broadchain graph FR assembly with a Broadchain. It's all. Thank you, everybody.
Can I ask a question?
Yeah, sure.
Sorry if I just misheard it, but do authors of your paper compared the Bruin graph algorithm with other algorithms of assembling genomes?
It's quite it's rather historical just things, because OLC, for instance, was used before the Bruin graph, and actually a Bruin graph uses the strategy of OLC, and where it uses in the not in the nodes, not in the comparison reads, but comparison of the graph. Here is a part of OLC. So a Bruin graphs hybrid between the Bruin graph and OLC.
Okay, thank you.
All right, so I guess no more questions. Thank you for your talk. And with that, let's go to the next one. Our last for today.
Yeah. Let me share my screen.
I'm sorry, can you see it?
Yeah, sure.
Hello everyone, today we're going to tell you about the Kano, which is the assembly tool for long read sequencing data specifically. So the long read sequencing, such as PacBio single molecule real-time sequencing, has the biggest advantage, which is obviously longer reads, which can be helpful in resolving long repeats. But the disadvantages are the higher error rate for the reads, and usually it's the lower depth of coverage. So addressing these disadvantages for the accurate assembly, what can we do about it? The authors of Kano propose that if we correct, trim, and properly filter our data, as well as accurately estimate the errors before the assembly, the results for long sequence data may be better. So the Kano implements 3 steps of assembly, which is the read correction, read trimming, and finally the assembly of a genome. So let's talk about them one by one. And starting with read correction, the read correction in Kano based on detected overlaps between reads, but the detection overlaps itself is kind of very computationally intensive procedure, and it's can be like 100 of millions k-mers in the human genome, and to perform all versus all alignment, you'd need the square of 100 of millions time. So the procedure utilizes the k-mer approach, but let me say in advance that the whole Kano pipeline is kind of revolving around reads, not k-mers. So for overlap detection, the reads are divided into k-mers, and for trespass this computational bottleneck of detecting overlaps, the min hash alignment process is utilized. So in Kano, it consists of 2 steps. First step is basically the identification of potential overlaps, which is performed with weighting k-mers with this TF-IDF approach. TF-IDF weighting, where TF stands for term frequency, which is frequency of k-mer inside the read, and IDF is inverse document frequency, which is inverse frequency of k-mer in the sequencing. An example is on the left. This is for first stage, and the second one is estimation of quality of the overlap, its length, and that is performed with a min hash bottom sketching procedure that illustrated on the right. So we have our reads, which are S1 and S2 on the top of the picture, that are broken into k-mers. And for each k-mer, we utilize some number of hash functions, which result in some hash number, integer number for each k-mer. And then we take the subset of k-mers with minimal hash value for each hash function, and that will be the subset of min-mers, and it is called the sketch of the read. And by comparing the sketches of 2 reads, for example, you can approximately estimate if there an overlap between them and how big it is. So the next part will be the read correction per se. Sorry for my paint artwork here, but the procedure is pretty simple. We already identified the overlaps, and for each read, we take some constant number of overlaps, and with them, which are, let me mention, which are weighted already. And for each read, we construct a directed weighted graph. And the graph with the maximum weight will be our corrected read. So on this step, we adjust the read sequence itself. And the next part is the trimming, which is pretty intuitive. So if the read sequence is changed, the length may be changed too. So the procedure is straightforward too. We just summon all the overlaps for one read, for every read, and look what parts of it are not covered by overlaps at all. And we delete these parts. So the read is trimmed or split according to where this unsupported part were. And the final step will be the preparation for building a graph and building a graph which is the assembly itself. So before building graph, we want to properly estimate the final error rate of our sequencing. So the main point here is that we just once again summon all the overlaps of all the reads and do not without changing the sequence of the read itself. We just look how each nucleotide is supported inside this read and write down this adjusted error rate after the correction and trimming. And the next step will be the some filtering for overlaps and the reads before we build a graph. So we have our recalculated error rate, which can be expressed as median error rate. And the overlaps inside the span of plus minus 6 median absolute deviance are considered as good and proceed into the graph. And another part for filtering is filter out suspicious reads. These are the reads which are not fully covered by these good overlaps. So such reads are probably chimeric or some repeats, and they will be marked as suspicious, and the procedure for them will be discussed somewhat later. So we have our good reads and good overlaps, so we can try to build a contigs with the best overlap graph, which has the reads as the nodes and the length of overlaps as edges. And at this point, there are also a need in procedure of placing the reads, including the suspicious ones, on this probable contigs. So for that, we estimate the error rate of overlaps over read, and such reads that are not exceed the predefined error rate margin are included into the subset which will be placed on the contigs. And then we also have the problem with the bubbles. And in the current state, the resolving of bubbles is that if the contig has some low quantity of reads, such as lower than the threshold for the number of reads, or it has more than 60 or 75% of overlaps with other contigs, that this contig will be considered as the bubble. And the reads that are placed on it will be kind of the algorithm will try to place it somewhere else. So basically, the final step remained is to build a consensus. So we have our contig, we have some reads placed on it, and building the directed graph, just as in the part of read correction, will give us the consensus contig sequence. And that will be all for the assembly. And as output, we will have our graph, our read layouts, and our contig sequences. So that will be it with methods. I think Margarita can proceed with the results of the simulations.
Yes, thank you. So all of these new modifications of approaches lead to overall improvements in runtime and quality of genome assembly. So authors of this paper tested it by comparing Kano with other assemblers in different tasks. Firstly, Kano is get better with repetition detection, and to test this, authors used the sequence of the Bacillus antarctis bacteria, specifically the Stern stain that doesn't have PXL2 PXL2, but has several copies of PXL1 plasmid. And so MinHash algorithm was compared to Delainer and Minimap. And as we can see, Minimap has the lowest positive predictive value, which means that the number of reads that were correctly mapped divided by the overall number of reads was the lowest, which means that there are more false repeat-induced overlaps. And Delainer actually has the best score, best PPV score, but it was memory and runtime not efficient compared to MinHash. So MinHash is the most optimal algorithm in terms of sensing overlaps. Next slide, please. Okay, thank you. So the next issue is better building of sparse overlap graphs. To test it, authors used simulated genome with 2 repeat copies that are like 100% identical. Then the graph was built. You can see it on the bottom left, and you can see it was built incorrectly. Here's a false overlap. And to test the sensitivity, again, the authors increased variations of repeats by changing single nucleotides in one of the copies. And as we can see, Kano was able to resolve the repetition at a smaller variation percentage compared to other algorithms. As you can see, only 3% difference lead to resolved genome, and other algorithms needed more variation to actually do the graph properly. And moreover, Kano was more memory and time efficient in general. Next slide, please. Okay, so now when we're talking about general efficiency of assembly, the authors tested the context quality and the time efficiency for PacBio and Oxford Nanopore data. So for PacBio, we can see that Canoe produces the most continuous assembly in most cases. The length of contigs is the largest. And in terms of the time, Canoe shows worse timing for small genomes, but here are two points. First, Canoe requires only one round of polishing, so it's like additional filtering of the data to achieve high quality. And other algorithms, for example, Miniasm requires up to 4 rounds to for this high quality, that leads to more time in total. And the second issue is that Canoe scales well to large genomes, for example, the human genome, like the last one on the table. And as we can see, it assembles this genome 3 times faster than Falcon. Next slide, please. And for Oxford Nanopore data, we see the similar. Oh,
Sorry. Yeah, sorry.
okay, so for Oxford Nanopore data, we see the similar results. In general, Canoe's assemblies were the same or more continuous than those of other assemblers. And again, Canoe shows a great timing in most cases. Next slide, please. So to sum up, Canoe is a great long-read assembly tool if it is particularly important to correctly identify and locate repetitions. For example, you need to recover a number of plasmids. If you're working with relatively low coverage data, and if you have limited computational resources. That's it. Thank you for your time. Do you have any questions?
I have a question, but it's maybe should be addressed not to you, but to our course instructor. As Mikhail mentioned, there was inclusion of specific to text method TF-IDF, and as a person who frequently work with text processing, I'm interested if there were some modification of this algorithm, because since TF-IDF was introduced, there were actually a lot of modifications that improved TF-IDF performance.
I think you should
Maybe
address
you
it
know.
to the authors, but as I recall, they just looked at formulae, which I presented, and they calculated directly the weight for each k-mer.
No,
there
I
is.
understood it. When this is why I addressed it's to course instructor. Maybe he knows something about this, because like if it is not, it is a area of improvement.
Well, it is kind of an older paper, I guess. Maybe people improved on that as of recently. Because, so yeah, Canoe is a relatively old tool, but it is used a lot, which is why I, well, presented it here for the review. I'm not sure about, maybe they use some other metrics, maybe more novel ones. So how new are these novel versions that are better than TF-IDF? Maybe they are
also pretty old, like for example, B25.
Mhm.
It is similar to TF-IDF, but it improved more in terms of context. You can read about this, basically also a formula, but more advanced.
All right, I have no idea actually. I might look it up to return to you. But yeah, not sure. I mean, it is possible that people who were working on Canoe didn't know about this novel things from text processing.
Pretty much, it's highly possible, but this is why I indicated that maybe it's a good research idea.
I mean, if you're interested in novel assemblers, you could try it yourself, I guess, implementing it if it is not implemented. It's not been implemented so far. But I can't give you an answer because, well, it seems like in assemblers, in some more recent papers, there is some room for novel ideas. It seems. Doesn't seem like everything has been done about it. So maybe if it is implemented, there are some papers. If not, I can't tell you why exactly not. Well, it's not really an answer to your question, but I might look it up. So if there are some assemblers, more recent ones that implement this idea.
I actually found the year of the paper of this new method. It's 2017. It's
So the
pretty
same
much
as
old.
this paper. But this
The
paper
year-
is from 2017.
Yeah, the year of the paper is also 2070, so
Yeah.
No,
So
I'm
and
not
it probably
arguing
was
that.
yeah,
It
it
is,
probably was written earlier.
Yes. It is interesting to find like application of newer methods. That's what I'm talking about.
Maybe there are some assemblers that are already employed. Maybe a newer version of Canoe uses it. Not sure. All right. So thank you all guys for coming. Thank you for great discussions today. And as far as I understand, we don't have any class next week because we have another module of system biology. So I guess with that, I'll see you guys in 2 weeks. Goodbye, everyone. Thank you all for coming.
Bye.
Thank you.
thank you,
Bye.
goodbye.